A very good morning to all the students and life members of Indian Society of Odontology. I welcome you to yet another brainstorming session of Rapid Review Revision course for postgraduate students. Today we have none other than our President, Dr. Nimpya Pandit ma'am. She is a professor and head of odontology at DAV Dental College, Yamunanagar, and ma'am will deliberate today on immunity and inflammation. Over to you, ma'am.
Thank you so much, Dr. Neeraj. You have been doing a great job of double role of being the treasurer as well as conducting all these, and you have been doing it so wonderfully. Thank you so much for being such a support. It's possible only because of you that webinars are going without any problem. स्क्रीन का ऑप्शन फिर से खोलनी पड़ेगी ना Okay. हेलो हेलो सर आपका अनम्यूट करना पड़ेगा.
Yes, ma'am. आपका आवाज आ रहा है.
हाँ. Okay, okay. Sorry about that. And so I was saying that it has been a great job by Dr. Neeraj that he's been conducting all this. And at ISP, we had planned a lot of activities this year, but unfortunately, because of the COVID, we can't do anything. So we are left with just some webinars. Before starting my presentation, I would like to tell you a small story about how why did I take this? Because this is one of the most dreaded topics. I remember when I was doing my PG, at that time, it was not mandatory to do library dissertation. And I don't know why, but the teacher, my guide, gave me this topic of immunity and inflammation. And once I went to the library, because those days the net was not available, I could found thousands of articles on that. And with the thing, I lost hope that I can ever write this dissertation. And sorry to say that because it was not mandatory for all of us to give the dissertations, library dissertations, I ended up not giving one. But then over a period of time, I wanted to face my scare and go about it. Uh, First, let me tell you that we are not immunologists, and so we don't need to go into so many details every day. New cells, new receptors, new cytokines, new these moieties of expression are coming in the immunology. We don't need to go into details. But I was looking at the question papers that have been asked in the so many years. A lot of questions have been asked in this from this portion of that, and because all other things like pathogenesis and your host response and your this host modulation, they are all related with this. So we can't escape it. Even if we don't like it, we need to know some basics about it and try to understand that. For your ease, I have tried to divide this topic into various portions, and I will be taking one by one. Although all of these topics integrate together to build up an immune response from the body. So the first thing what comes to our mind in the immunity and inflammation is, what is the relation? Why do we say immunity and inflammation? Why is it separate? Why is inflammation separate from immunity? It's not separate from immunity. Inflammation is the first response. So you can easily say that inflammation is same as innate immune response, which is the first response of the body to anything that is external. That may be a chemical, microbe, or heat insults, or any insult to the body. This is the first response. So inflammation is basically the innate immune response, which is not specified towards a microbe or towards a particular stimulus. It's the first response. So inflammation, you can write up to the areas where the innate immunity stops and the adaptive immunity goes. So inflammation is those processes you must have studied in your second year and third year, that there are 5 signs of inflammation, and what are the reasons for that. Those you can write about that, because questions have been asked about inflammation also. So once the first response of inflammation is there, and the body tries to take care of that, whether it's the injuries are heat, chemical, bacterial infections, anything. And if the body is not able to handle the infection or the external stimulus at this moment, then the body goes into the era of chronic response, which is the chronic inflammation, which is the most common a disease that we found in the periodontium, that is gingivitis and periodontitis. So these are chronic infections that we are studying about. It's not acute. Acute inflammation will be only like if you're talking about abscesses or pericoronites, those are acute inflammation, and they are usually seen with pain and inflammation and some discomfort in that area. And we know that one of the basic things about periodontitis is that it does not lead to a lot of pain and discomfort to the patient. That's why the patient doesn't come to you for a long time. So over a period of time, we have seen that if we relate this immunity and inflammation to what we have already studied in the 4 stages of how the gingivitis progresses to periodontitis, we have the initial lesion, where it's an acute injury, where it's the first response or vasculitis, and this portion you can say is the inflammation. Then second is the early lesion, when there is a dense infiltrate of T lymphocytes and pathological alterations of fibroblasts. The fibroblasts start elaborating the collagenases or MMPs, and then the collagen degradation takes place. The third is the established lesion. It's still gingivitis, where we have not still progressed to the periodontitis. There is predominance of B cells, and there's a further loss. There is further loss of the matrix, further infiltration of the cells, further swelling of the tissues, and ultimately, once it crosses that barrier of junctional epithelium, it goes to the advanced lesion, that is our periodontitis. So in the periodontitis, the last stage will be the B cells will predominate. So we start with the neutrophils, go to the T cells and B cells, and ultimately when the B cells predominate and the inflammation goes wayward, it is not controlled by the body, then we say that it's an advanced lesion or it's periodontitis. So these are the 4 stages which you can correlate with the immunity or inflammation. Now this article, I think all of you should go through this, because once we start thinking about inflammation and immunity, we always think about so many comorbidity factors. For example, genetics or your metabolic diseases or the hyper-responsive monocytes. Then you get confused. You feel like that whether we should go about saying about only how the cells gradually change and how the situation changes in the local tissues. We also are concerned about what are the implications of other factors. For example, genetics. For example, you have hyper-responsive monocytes. So all these things in this article, which was given by Goldberg in 2016, he said that that it's a dysregulation, because we all of us know that it's not a external infection; it is the dysbiosis in the local tissues. Immunostat is how sensitive is your immune system. Should it be, if it's oversensitive, that also is a problem. If it's undersensitive, that is also a problem. Hypersensitive, it will lead to infective disorder. So this novel concept was given, which will incorporate the other factors that are affecting the inflammation or the immunity in the local tissue. So taking these into considerations, because we cannot go into details, there are of proteins. Those proteins ultimately lead to autoimmunity. They make our own defense cells active against our own cells, and so there is more destruction rather than the repair and the elimination of the microbe by those things. So we should concentrate upon the things which are going in a normal way. Then we should try to understand what are the other additional factors which may lead to over responsiveness of that. So this article will give an idea about how the other factors fit in this pathogenesis of the periodontitis. Now this is again a very complex type of a diagram, but try to understand this, how the other factors can be incorporated in this. First, we were trying to see that it's an infection. That was the area when we started making vaccines. We thought it's an infection, like we are looking for a COVID vaccine right now. So we were looking for a vaccine against P. gingivalis. But you know that the research is not going more in that direction, because we know now that it's most important thing is that it's not an exogenous infection. All these bacteria are already in the oral cavity, and those are actually going from symbiosis to dysbiosis. They are not, that has already been taught to you over a period of time. So in this way, we are looking at the host response from a renewed interest, and we try to understand what are the steps why from this. Why do we need to go into the details of those steps is that we could understand at some point that we can stop and we can intervene and stop the periodontal infection into going into an era where it is uncontrollable. So immune response basically also includes the strenation, what I was telling you, that the body goes into autoimmunity mode, and then there is no way you can stop it, because if the body is sensitized towards its own cells, then it's a difficult situation to control. Now I'll take this lecture in the following ways, because you have a lot of questions in different aspects of this immunity and inflammation, and we will go about the contents are complement, neuropeptides, toll-like receptors. You must have read about all of these, but let's make it in such a way that you can write answers, at least the short notes related with each one of these. So I'll go about like this. These are the different. I would suggest you to go through this article. I was telling you that it was a very difficult task for me to write a thesis on immunity and inflammation, but recently I found out this article, and I think if you go through this article and see the different subcategories in this, you can very well write the answers related to that. So this is one of the article I would recommend you, all of you, to go through. So if we go about, let's make a story out of this, how the immune system works. Now, first, you know that at this clavicular level, there are different types of antigens from the bacteria that somehow gain entry. They pass the epithelial barrier, they somehow gain entry into the connective tissue, and these are the different factors that will act. We will go in detail about all of these factors, but if we try to follow it like a story, you will understand at each stage, if one mechanism falls, then another mechanism takes over, another mechanism falls and another mechanism takes over, and ultimately it leads to the bone loss. Now you would think that bone loss is actually a protective response in a way, because bone loss will take the main tissue away from the inflammatory part. So bone resorbs in a protective way, that the body does not, the infection does not go into the systemic circulations. So this is also over all the slides, you will know that this starts from the epithelial barrier, innate immunity, resident cells of macrophages, and then T cells and B cells, antibodies, and ultimately there is bone resorption. Now, our current understanding says that the immune system has two arms. One is innate immunity, adaptive. Although I would say that these two systems are not separate from each other. If adaptive immunity is there, innate immunity is also working at the same time. They are overlapping. We are just dividing these because we need to understand the two aspects of it. Otherwise, if adaptive immune system is there, there is innate immune system is not shut down. It's still working. So what is innate immunity? It's an instant, immediate, and it integrates with the adaptive system. So this part, you could say, is the inflammation part, and adaptive immune system is the chronic inflammation. So looking at the cells, just let us revise that stem cells, they are divided into lymphoid cells, which can further be classified into B progenitor cells, T progenitor natural killer cells. T cells can be Th cells and T cytotoxic cells, T helper cells and T cytotoxic cells. B cells are, this is the humoral immunity, this is the cell mediated immunity, and these are the natural killer cells, which are a part of innate immunity. These are a part of adaptive immunity, but this is a part of the innate immunity. And from the granulocytes, we have neutrophils, which are the first line of defense. Although I feel that if you have resident macrophages, they are the first line of defense, and they only recruit more neutrophils in that area. We have the basophils, mast cells, monocytes, dendritic cells, and macrophages. So this is the classification. If we look at the actions of each monocytes, now you need to understand two things. There are DAMP and PAMP. DAMP is damage-associated molecular patterns, and PAMP is pathogen-associated molecular patterns. These are two things which are a threat to the body. Damage-associated molecular patterns can also be seen in the cells which are dying, and they are producing some some antigens which need to be cared for, because these are the damaged cells. The body needs to get rid of these. So these are damage associated molecular patterns, and EAMPs are the pathogen associated molecular patterns. Or MAMP is another term which you need to know, is microbe associated molecular patterns. Pathogen associated molecular patterns and microbe associated molecular patterns are almost similar. So all these cells, if there is a damps, they will lead to the recruitment of different types of cells. Out of these are the innate immune cells, monocytes, dendritic cells, neutrophils, mast cells, natural killer cells, eosinophils, and adaptive immune cells. They are the T cells and the B cells. But initially, when we were studying immunity and inflammation, we thought there are only innate immune cells and adaptive immune cells. But over a period of time, we know that now other cells, non-immune cells, are also participating in the immunity. These are the epithelial cells, endothelial cells, and the fibroblasts. They also elaborate cytokines. They also recruit different types of cells, and they also elaborate enzymes which are important for the degradation or the healing of the tissues. Fibroblasts are important. Again, if they elaborate collagenases, they're important for the degradation. And if they elaborate some the collagen, they are important from the healing point of view. So it's not only the innate immune cells or the adaptive immune cells which are important. We also have non-immune cells which participate in the defense mechanism. So what are innate immune cells? They are the cells of non-hematopoietic origin. That is epithelial cells. That is the first line of barrier, what I was showing you in the last slide. Of the myeloid cells, that is phagocytes, those are the B cells and the dendritic cells, innate humoral defense and complement casting. We'll study these one by one. So what is innate immunity? It is the first immunity. It's non-specific, characterized by phagocytosis and digestion of microorganisms and foreign substances by macrophages and neutrophils. These are the two main phagocytes which are present in the body, which will ingest the foreign material. So how does it do it? The macrophage elaborates on its surface a receptor, and it's attached with an antibody, which we'll study in detail, and it attaches to the antigen which is found on the bacteria. And this may either engulf it and kill it, or it may present it to the T or B cell. So this will study. I'll show you a small video about how the things go. Then we can follow these steps more in a better way. So we'll just look at this video.
Nearby bacteria seize on the opportunity and enter your world. They start using the body's resources
मैम वीडियो प्ले नहीं हो रहा है.
हेलो,
वीडियो प्ले नहीं हो रहा है.
प्ले नहीं हो रहा है.
नहीं. फिर से प्ले.
वीडियो प्ले नहीं हो रहा है. वीडियो प्ले हो रहा है, कह रहे क्लियर नहीं है शायद. नहीं, प्ले नहीं हो रहा.
यस मैम.
अब हो रहा है?
यस यस मैम.
अब हो रहा है सर?
यस यस मैम.
ठीक है.
मैडम उसका ऑडियो नहीं आ रहा है।
हाँ,
ऑडियो
ऑडियो नहीं आ रहा।
नहीं नहीं।
ऑडियो नहीं आ रहा। अभी तक आ रहा था सर?
नहीं मैम, वीडियो उसमें नहीं आ रहा है, ऑडियो आ ही नहीं रहा है।
Nearby bacteria seize on the opportunity
अब
and
आ रहा है
enter
सर?
your
यस यस मैम।
wound. They start
आगे चला लो, आधा आ रहा था।
यस मैम आ रहा है, अब आ रहा है। नहीं फिर से गया है मैम, वॉल्यूम फिर से ऑडियो गया।
ऑडियो फिर से गया, पहले आ रहा था।
हाँ अभी आपने शुरू किया वीडियो तब आ रहा था।
पहले आ रहा था फिर से गया। फिर मैं इस पे बता दूं, ये रहने सर, कोई बात नहीं, आई विल स्पीक आल्सो।
यस यस मैम।
हाँ, आई विल स्पीक along with the video. So the bacteria, they divide within 20 minutes into millions of bacteria. And the first line of defense that they find is the macrophages. The immune system has to stop them as fast as possible. There are macrophages, resident macrophages, which first come into action. And they're the biggest cells. They have a diameter of 21 micrometers. But there are millions of bacteria, so they suffocate the attack. That is how the gingivitis progresses, because they overwhelm the immune system, and they either engulf some of these bacteria and engulf them, and then they send messenger proteins for the recruitment of more cells, because it's not contained by the macrophages. Then cytokines or the messenger proteins are released, and they recruit the neutrophils to the area of infection. Neutrophils fight the toxins and they try to neutralize the toxins, and they also have extracellular nets, NETs. You will get a question on that, on NETs. And over a period of time, because it's overwhelming response, they get killed over a period of time. And then they recruit the other cells, which are the dendritic cells, which is called as the brain of the immune system. Then the dendritic cell will divide this antigen into various and express it on the surface of the dendritic cell. Then it will try to see whether this is a bacterial or a viral infection. And then it finds that if it's a bacterial infection, it will recruit the same type of cells. They will travel to the local lymph nodes, and they will stimulate the same colony of T lymphocytes, which will divide over a period of time, and there is an army of T lymphocytes which is formed. And the dendritic cell with its antigen on the surface will try to find the same type of T cells which can bind the same type of antigens. And once the same type of antigen antigen and T cell reaction takes place, there's a division of the T cell per cells, and huge cells are produced. Some of them go into the memory, because if second time there is attack, then they will have a more bigger number to first take care of them. The T cells will activate the B cells, and cells will rapidly proliferate. there is actually, they produce a lot of antibodies, and macrophages will come into picture, and they will identify the different types of antibodies on that. They will bind and they will try to phagocytose them. So this is a sequence of events that has been seen. Now I'll go to the main presentation. Now we'll study in detail. This was just a sequence of events I wanted you to go through, so that when we are going through the slides, you will know what are the sequence of events. Now, one by one, first we'll discuss about the complement. Complement, the name itself says that it complements the ability of antibodies and the phagocytic cells to kill the microbes or the damaged cells. We talked about the DAMPs and PAMPs, so we are talking about microbes as well as the damaged cells. Immune system is also responsible for taking care or eliminating the damaged cells in the body. It promotes inflammation and attacks the pathogens plasma. So why do these complement system, what is it? It's a system of almost 25 proteins which affect almost all the immune cells of the body. So there are different components of that. It's a C1 to C9 components are there, and they are involved in the opsonization in the immunity, lysis of pathogens, chemotaxis, inflammation, cell activation, clearance of immune complexes, because they bind to different types of the antibodies, and then they clump together different types of antigen antibody complexes, and they eliminate it. And adaptive immunity, they augment the antibody response, promote T cell response. So it's a system of 9 proteins. Now we need to go into detail. There are 3 pathways by this system is activated. All these proteins are found in the blood in the inactive way, and there are different ways by which these pathways can be activated. So the first is the classical pathway, that is antigen antibody reaction. On the surface of if there is an antigen antibody, we have already seen in the video that there is a binding of antigen with the antibody, and the pathogen surfaces are there. So what happens is that there are some proteases which will cleave C1 and lead to the C1 and CR and S. And this will cleave further, this will become a protease, and it will cleave C4 into A and B, and it will cleave C2. And C2AB, C4AB are basically C3 converters. That is, they will convert the inactive C3 into C3A and C3B. There is another pathway which is called as the mannose binding lectin pathway. What is mannose binding lectin pathway? That there are some bacteria which have a mannose binding site on their surfaces. So this mannose will actually bind onto the surface of that pathogen, and this associated with the other 2 proteins, MASP and MASP2, which are same as C1 and C2 are, they will again cleave C4 into A and B and C2, and ultimately lead to the formation of C3 converter. There's an alternative pathway. Although we see in periodontitis that antigen antibody reactions are there, but when we investigated, it's the alternate pathway that is most active in the periodontitis. So how does it activate? It is by the lipopolysaccharides on the pathogen surface, that they directly cleave the C3 into CA convertase. And the A's and B's, you should understand that C3A and C5A, they are basically the anaphylatoxins. That is why the A, you can remember the name by A. These are anaphylatoxins, and they are the main portions which are important for the inflammation as well as the release of histamine and anaphylactic-like reaction in the hypersensitive host, or they are the mediators which will recruit the different types of cells in the site of inflammation. C3b is a part of the membrane complex. Now C3b will bind with the C5b, 6, 7, 8, and all these proteins will form. They will recruit C9 proteins from the plasma around that cell, which we need to kill. Now we basically need to kill the pathogen. what will this complex do? There are C9 proteins in the plasma. They will bind those, and then this is a pore forming area. They will form a small pore in the membrane of the pathogen, and it will ultimately lead to the lysis of the pathogenic cells. So we should remember that it's the alternate pathway which is more important from the periodontal point of view, although these two pathways are also working there, and C3 convertase is the main enzymes which ultimately leads to the formation of the membrane. And C3b has other actions like opsonization. Opsonization, you must be knowing, is that if there's a low molecular weight pathogen which is not recognized by the body as a threat, then these small portions of C3b will attach with that, increase the molecular weight so that the body recognizes these as something foreign and takes care or gets them eliminated from the body. Now, another thing is neuropeptides. You may not have heard about this because it's a new addition to the immunity and inflammation. You have heard about the different types of inflammatory mediators like kinins, histamine, prostaglandin, eicosanoids, and other things which recruit the different type of hormone. But recently in 2016, there was a study which said that even the nervous system is involved in the inflammation. It is the neurons which generate the electric impulse, and they will decide whether this inflammation should go into the chronic or it should be stopped. So alternatively, now we say that neurons will elaborate neuropeptides. There are other cells also which elaborate neuropeptides, and three main neuropeptides that are important from the periodontal point of view are the substance P, vasoactive intestinal peptide, and this calcitonin gene related peptide. So these are three things which have been seen. Why we are studying them, I'll tell you. So there was this study, I would like you to go through that neuropeptides and neurogenic mechanisms are there in the periodontal inflammation. So what are these neuropeptides? They are secreted from the nerve cells. This is calcitonin gene related protein, substance P. These are important, and vasoactive intestinal membrane. What happens is that once the noxious stimuli is there, the mast cells elaborate histamines. Now this histamine is taken up by the cell body of the neurons, and they elaborate different types of substance P. So they regulate, either they enhance the inflammation according to the local whatever is requirement, or they decrease. They're also important in the way that we say that histamines and this bradykinin, they lead to the arterial dilatation and extravasation. These substances also increase the local dilatation of the blood vessels in that area, and they will influence the immune system. Not only can they lead to the different responses from the different cells, they also, neuropeptides are also released from neurons. They play important part in the immune cells. Now immune cells also have receptors for these neuropeptides, different types of neuropeptides, and accordingly they will form different types of cytokines which will either enhance the inflammation or which will regulate the inflammation. There is one receptor which is important from the periodontal point of view, which has been studied over a period of time. This is the protease activated receptor 2, which is found on the nerve cells. So this nerve cells, once the microbe is there, this will lead to the elaboration of different intracellular events, which will lead to the cytokine formation and increase in the inflammation. So this is one way of how the neurons are activated during the inflammation. This protease activated receptor is co-expressed with substance P. I was telling you there are three these neuropeptides which are important from the periodontal point of view. So they're expressed with the substance P and where it's believed to mediate the inflammation. So this is how it will activate with the substance P and lead to the inflammation. Now, neural growth factor is another factor which has been shown to directly regulate the synthesis of calcitonin gene-related peptide by B cells, and it does in the sensory neurons. Now, this is another component that has been added apart from the inflammatory mediators, which are usually the lipid mediators or the kinin mediators. This is an added thing which has been, this also explains why we have more periodontitis in the stress patients, although that is not a one-to-one relationship, but this may explain another mechanism by which the nerve cells are contributing to the inflammation. So what are the periodontal implications of these neuropeptides? What we have seen that in many experiments, we have seen that the levels of substance P in the gingival tissues are reduced after periodontal treatment. That means it was elaborated in more quantity in the gingivitis patients. and in the periodontitis patients. Calcitonin gene-related peptide has immunosuppressive action. So this one is enhancer of the inflammation. Calcitonin gene-related peptide is the suppressor. So this is a more of a regulator. And vasoactive intestinal peptide is more of a pro-inflammatory and anti-inflammatory. If the microbes are not cleared from that area, it will have a pro-inflammatory action. If microbes are cleared and you don't want the body to go into more damage mode, then it has an anti-inflammatory mediators. So this one is enhancing the inflammation, this is suppressing the inflammation, and this is both actions depending upon what the local meaning is elaborating over there. Now this is kinin system. You must have read about that. These are the inflammatory mediators, and bradykinin is released from the local mast cells and histamine, which leads to pain, vasodilatation, vascular permeability, and chemotaxis. So which are together along with this and neuropeptides, they are taking care of the initial inflammation. So you can write this in the inflammation part of it. Next is toll-like receptors. This is one thing which needs more attention because a lot of questions have been asked from this, and you need to understand how it elaborates into the different types of responses from the body. One of the primary challenges of the immune system is to differentiate the host from the bacteria. So how it is done? It is done with the PRRs, pathogen recognition receptors. So what are they? Now differentiating self from the non-self. I was telling you PAMPs, pattern recognition receptors, they take care, and then if the body, if it's a self marker over there, the immune system will not react. If it has a surface marker which shows the different configuration as the body, then it will lead to the activation of the immune system. So after recognition of microorganisms and foreign substances, chemokines are secreted to attract the pathogens. Now, this is how it recognizes. This is about the PAMPs and this is about the DAMs. So there are two threats to the body. One is pathogen-associated molecular patterns and damage-associated molecular patterns. So these are recognized by the body, and this leads to the elaboration of cytokines and chemokines and inflammation, adaptive immunity. And ultimately, once the immunity takes care of all the foreign substances or the damaged substances, then the tissue repair are also elaborated by the cytokines. That is also interesting. On the basis of function, we divide them into these pathogen recognition receptors into signaling and endocytic. Now, here you need to know that signaling ones are the ones which directly recognize some patterns on the pathogens and the viruses. This one is on the surface, and NOD-like receptors are found inside the endosomes. They are associated with the membrane. And there are some C-like lectin receptors, which are important from the fungal point of view. So the stall-like receptors are important from the bacteria point of view. Nod-like receptors are important from the viral point of view because their virus gains entry inside the cells. And these are important from the fungal point of view. So you have three types of receptors to take care of three types of the threats to the body. Now going into more detail. So this is the C-like lectin receptor, toll-like receptor, and in the endosome, on the surface of the endosome, it's very easy to understand that this can take care of the fungi, this can take care of the bacteria, and this can take care because the virus will gain entry into the body, then there has to be something inside the body which will recognize that virus and ultimately lead to the elaboration of cytokines. This ultimately leads to the formation of kappa proteins, and this will lead to the formation of IL-1, IL-6, which are pro-inflammatory mediators and which will take care of the inflammation later on. So again, we have different types of these toll-like receptors. I will give it the classification of that. From the periodontal point of view, TLR4 and TLR2 are important, and from the viral point of view, it's the 3, 7, 8, and 9, which will recognize the viral and bacterial. Nucleic acid, if virus is there, it will be recognized by these and ultimately lead to the transcription and formation of the cytokines. Now now you need to know about the oligomerization domains. There are some parts inside the cytosol which will bind together, form different proteases, and ultimately lead to inflammation. So these NOD1 and NOD2 are important from the inflammation point of view. Now, first, let's go through all these toll-like receptors. There are different types of receptors. 11 have been seen, and you can see that this is intracellular, 3. These are all on the cell membrane, and you can see that P. gingivalis actually is important for the TLR2. It stimulates TLR2 and it also stimulates TLR9. So this is one organism which has a receptor for at 2 TLRs. So you have different types of receptors for the different types of microorganisms or virus or fungi. Now, how does this take place? First, the pathogen invasion is there. If it is the surface of the cell, then this is the TLR. It will engage this TLR. This TLR will engage the pathogen. And ultimately, there is an activation of intracellular pathways. You need to know that there are two types of intracellular pathways by which cytokines are made. This is myeloid differentiation 88 and IRAK. IRAK is basically for the interferon type of activation. And myeloid D88 is for the rest of the bacterial cytokines. And there is activation of some kinases once this recognizes the antigen, and there is release of nuclear factor kappa B, which will ultimately lead to the activation of CD20 and TCR and activation of the T cells and elaboration of cytokines and activations of the T cells. Now, again, going from the, we have studied about the complement, we have studied about the toll-like receptors. Now, let's go into this journey, what I was showing in the first slide. Let's go through the journey of how the microbe gains entry into the deeper tissues and leads to the inflammation and chronic periodontitis. Now, this epithelial barrier is the first barrier which will try to stop the microorganisms, but this is not helping. In the epithelium, we have dendritic cells. Once even the saliva, you know that there are antibacterial factors in the saliva, they will try to contain the microorganisms. And the first epithelium I was telling you, it was not considered to be a part of the immune system, but now we know that it does elaborate. Some antimicrobial peptides are elaborated by the epithelial cells and they will try to contain the organism. But the organisms will further grow. What happens to the dendritic cells? Inside the epithelium, the dendritic cells will lose its cohesiveness with the surrounding cells. They will go to the local lymph nodes and they will activate a particular type of T lymphocytes and B lymphocytes, which will multiply and take care of the pathogen. So they will gain entry through the lymphatics into the local lymph nodes and they will try to elaborate or try to stimulate a particular colony of T lymphocytes, which will ultimately lead to the, which will be elaborated in the blood and take care of the pathogen, which is not actually stopped by this epithelial barrier or by the antimicrobial peptides. Now, there are, on the basis of spacing and pairing, there are two types of defensins that have been seen, alpha defensins and beta defensins. And this alpha is produced by the PNMs and the beta defensins are produced by the epithelial cells. Now, these are different. They attract dendritic cells with beta defensins and they ultimately will lead to the adaptive immunity. And they will also function as an enhancer or binding between the innate and the adaptive immunity. Inflammasomes. You have a lot of questions of inflammasomes. Let us try to understand what this is. What I was telling you till now, that there are MAMPs and DAMPs which will try to have intracellular events. Now, what is an inflammasome? It is the receptor, nod-like receptor, an enzyme, caspase, and adapter protein, and the combination of these three is called as inflammasomes. So what is an inflammasome? If a bacteria or any type of foreign body comes in contact with that, there is a complex of different types of enzymes which will collaborate together and they will form the inflammasome. So ultimately these will lead to the kinases which will cleave some parts and activate some areas, and ultimately they will lead to the elaboration of formation of nuclear kappa factor B, and there will be elaboration and the formation of different types of cytokines. So this is inflammasome is nothing different. It is just a combination of different types of proteases which work together for the formation of cytokines. Now, these are different types of inflammasomes. They are NLRP, NLRP 1, 2, 3, and 4 are associated with the physiological function, with the normal activity of the body, and they produce IL-1, 8, L33. These inflammasomes will stimulate the formation through the nuclear kappa factor. Now, this is about the complement neuropeptides, toll-like receptors. Now, we'll go to the antigen presentation and activation of the acquired immunity. What is adaptive immunity? It refers to the antigen specific. We were now talking about the non-specific one. Now this is something called as specific. This is specific because now the dendritic cells have lost their cohesiveness. They have come to the local lymph nodes. They have activated a particular colony of lymphocytes, and those have divided in large numbers, and they will try to neutralize or kill the bacteria. So now at this stage, when the body goes into the acquired immune response, this is actually something which has gone beyond the control. This has crossed, antigen has crossed the epithelial barrier. It has not been contained by macrophages, by neutrophils, there are immune complexes which will ultimately lead to the formation of different cascade of events in the complement, and then there will be osteoblastic or osteoclastic bone resorption by the rankle production. We will go into detail of that. So how is antigen presented? You need to know about the major histocompatibility proteins. There are two types of MHCs, major histocompatibility complexes, which are found. One is found in all the cells of the body apart from the cells which are the immune cells. MHC2 class 2 is found only on the immune cells. So it's very easy to understand that MHC class 1 will take care of the damage associated molecular patterns, because these are found in all cells. And MHC2 will take care of the pathogens, because now these are immune cells and they have to phagocytose the bacteria. So if these are activated, then they will lead to the damage of the cell, because this cell is damaged and it's again elaborating toxins, and MH2 is associated with the antigen antibody complexes. So there are some autoreactive antibodies, and they include now these cells which are damaged in the body and which are associated with the class 1 and beyond, they have to be taken care by the cytotoxic T cells. So these T cells will take care of the cells which are damaged in the body, and MH2 are basically found in the suprabasal tissues. Now, what is the difference between two types of MHCs is that MHC is present in what I told you in all the cells, including the platelets, and they are only found on the dendritic cells and antigen presenting cells. And these are the different genes that are encoding for them. And presentation is of the endogenous origin. I was telling you the cells which have been damaged, they will be taken care by the MHC class 1 elaboration, and the ones which are the extracellular proteins which will be taken care by the MHC, because these MHC class 1 and 2 are elaborated on these cells, on the cells of the immune cells, and then only they will be recognized by the phagocytes like neutrophils and macrophages, and then they will be eliminated from the body. So once the microbes are there in the surrounding areas, and antigen presenting cell takes in the microbe and it elaborates these antigens on the surface, there is differentiation of the T cell into Th1 and Th2, helper 1 and helper 2 cells, and these cytotoxic cells, as I was telling you, will take care of the cells which are damaged in the body. And Th2 will actually function in relation with the B cells and elaborate different types of, it will become in a blast stage and ultimately produce antibodies which are specific towards that particular antigen. So this is the sequence of events, how the antigen presenting cell will differentiate different types of T cells, and ultimately B cells lead to plasma cells and will lead to the formation of antibodies. Now there are cell mediated response, that is the T cell responses, which are basically, there are two types of cells which have on the surface CD4 receptor and CD8. CD4 is the helper cell and CD8 is a 4. So CD4 will be with MHC2 and CD8 will be with the MHC1. So immune responses are regulated by difference. There are different types of studies have been done. Some say that Th1 hyper response will lead to more of periodontitis. Some say that Th2 type of response will lead to that. What decides which type of cell will be more effective is that the type of cytokines that are present. We'll study about the cytokines. So the cytokines are the deciding factor whether the Th0 cell will go into Th1 or Th2 cell, and whether there will be overwhelming response or the response will be contained. The other types of cells that have been seen with the T helper cells are the Th17 and T regulatory cell. This is Th1 and Th2, and the different type of milieu that is IL-12 and interferon will lead to the differentiation into Th1 cells, and IL-4 and IL-2 will lead to the differentiation into Th2 cells. So this is important from the antibody mediated reaction, and this is from cell mediated type of reaction, autoimmunity and inflammation. Pre-regulatory cells are found, they have been recently found, and they are important from the immune tolerance point of view, that they down-regulate the formation of more of the T cells or the cell type of mediator. Th17 cells, they elaborate IL-17, interleukin 17, 21 and 22, and they are more pro-inflammatory type of cells. You have Th11 also, or IL-11 also, which is called as an anti-inflammatory, and IL-17, which is pro-inflammatory. So the balance of interleukin 11 and 17 will decide whether the disease will further progress or it will be contained at this particular level. So how do the regulatory cells in the periodontitis, these Th cells, how do they regulate different type of differentiation? So these regulatory cells will not allow the osteoclast precursor cell to go into the osteoclast. So this regulates the formation of osteoclast and ultimately leads to the stopping of the bone loss in that particular area. Now, role of Th cells is that it secretes, as I was telling you, IL-17, 22 and 23. And it's thought to be important in the pathogenesis of cell-mediated immunity, and it's also important from the autoimmunity. This is the cell which will ultimately lead to the uncontrolled elaboration of different types of cells. So Th cells is also expressed in the Wrangler type. Now there is a T cell suppressor activity. Suppressor activity is that if the T cells go on dividing, then there will be formation of a lot of cytokines, and they will damage the host tissue more than the organism. So there are the T suppressor cells which will try to downregulate these different types of cells and not lead to the formation of more cytokines. Now this is the pattern how T cells are elaborated. This is the pathogen which is taken up by the dendritic cell. It is elaborated on the surface of the antigen presenting cells in collaboration with the MHC2 proteins. These naive T cells attach with that and they will differentiate into different types. Now, if the body does not contain the infection by the T cells also, now the body mode, it goes into the humoral immunity or the FRAC periodontitis, what we call as an advanced periodontitis. At the periodontal sites, we see that there are both locally produced antibodies as well as systemically produced body. The first antibody that is produced is IgM, and later on there is antibody switching to IgG antibodies. So on the surface of the mucosal surface, pathogen-specific Ig is local. We have secretory IgA, IgG, and systemic IgG. So how does the local production of salivary or secretory IgA take place? There is a microbe which gains entry into that, and it leads to the plasma cell formation, and these are the formation of IgGA, which will be elaborated on the mucosal surface, and this will take care of the organism. But this is a part of the innate immune response. But the formation of specific antibodies towards antigen is the part of the specific immune response. So if in several autoimmune diseases, we see that there is an increased production of IgA, and they keep on elaborating on the epithelial surface, and there is epithelial damage in those. So this is a part of the autoimmune system where there is more elaboration of IgA on the surface of the epithelium. Now there are two ways by which T cells will stimulate the B cells. Either they will directly, either the antigen will directly stimulate the B cells to form the antibodies, or they will stimulate the B cells through the T cells. So this is T independent antigen, and this is T cell dependent. If the antigen is dependent on the T cells for the formation of antibody, then we call this that. So most bacteria in our periodontitis, they present that is a T cell dependent antigen. So what happens by how does it activate the B cells is that pathogen is first ingested by the antigen presenting cell, then the proteins are digested and they are attached to the class 2 MHC proteins. The complex is moved outside the cell, and then the helper cells, T helper cell recognizes and improves it, and activation leads to the formation of cytokines, which ultimately will lead to the T cell, B cell proliferation maturation. So you can have a T cell dependent activation of B cells, and you can have T cell independent activation of B cells. This is another diagram, antigen presenting cell, activated T helper cell in elaboration with MH2 proteins. B cells are there activated, IgM, IgE and IgA are produced by the B cells. Now this is an independent mechanism. What happens here? You don't need a T cell, you directly the pathogenic bacteria will associate with the B cells, which will lead to the formation of the different types of antibodies. You don't need to have T cell dependence on that. So this is a T cell independent B cell activation. Now this is a stage which you need to know. This is a progressive stage or an advanced stage. These microbes, they have not been taken care by the local antibodies. They have not been taken care by the macrophages or neutrophils, and they ultimately have led to the formation of. Now look at these antibodies. They are not very specific. So this is a stage where we say that there is a uncontrolled elaboration of antibodies. These antibodies, some of them will bind to the antigen and neutralize it. And there are some antibodies which are non-specific, and this is called as non-specific polyclonal B cell activation. Many production, but now these antibodies are there which are not doing their job, but they are only contributing more to the formation of cytokines and ultimately to the destruction of the local tissues. Now numerous studies have been carried out that elevations in human immune are associated with increased severity of disease. That is what I was telling you. There is a polyclonal B cell activation, uncontrolled formation of the different types of cells and elaboration of many types of cytokines, which will lead to the formation of destruction of the periodontal tissues. Now, there is another thing which you need to know is that isotype switching. That is what I was telling. This is the biggest type of, this is the largest molecular weight, IgM. So first, this is the first type of immunoglobulin that is formed. Now, over a period of time, T cells will change this antibody to the IgG receptor, which is more easily taken up by the phagocytes. If it's an IgG, it's switching from IgM to IgG. This is easily recognized by the phagocytes, and this will lead to the elaboration or the killing of the bacteria in this local region. So this is what I was telling you, polyclonal expansion of the B cells. And one of the important factors for polyclonal is LPS. So this is a mechanism by these organisms, these gingivitis particularly, that it leads to the elaboration of non-specific antibodies which will not do their job and only cause more recruitment of cells which will be taken up as nutrients by the microorganisms. Now, what is the sequence of events for the formation of antibodies? These are the antigens which will lead to the cell proliferation. There is clone of plasma cells which will release antibodies into the circulation, and another clone is there for the memory cells, because another time the attack occurs, it will have a quick response rather than waiting for the these different types of microbes to have a particular colony activated. Now, these are the different functions of the antibodies. It enhances the phagocytosis because it attaches to 2-3 bacteria and it agglutinates, and it is easier to eliminate this bigger clump of bacteria. Opsonization, they will form on the part and then they will be taken up by the phagocytes. They will cause lysis by activation of the complement antibodies, and they will cause inflammation also, and cell mediated immunity, and there can be neutralization because they are again binding, and they are binding on the toxin part. So they are actually hiding the toxin part, and it will not be, no more be harmful for the body. So these are the different actions of the antibodies. Now, another thing which is important from the periodontal point of view is the FC receptor. Over a period of time, we have seen that there are many studies which have been carried out on the FC receptor. FC receptor is found on the effector cells, and there have been studies. I'll tell you the study. There are 3 types of main types of FC gamma receptors, 1, 2, and 3. In a Caucasian population, we saw that the FC gamma receptor 3 was having, in moderate and the mild type, there was no difference in the FC gamma R1 or 2 receptors. But in the advanced bone loss cases, there was a significant genotype association with the FC gamma receptors. So you need to know about this also. Now we go to the cytokines and chemokine networks. What is the difference between cytokine and chemokine? Cytokine is a broader term. Chemokine is that cytokine which has a attack and property which actually has a chemical gradient which will migrate different types of cells to the local areas. So this is an array of different types of chemokines and cytokines which have been seen in the periodontal tissues. Now, cytokines are actually elaborated by all type of cells. I was telling you the stimulus to any type of cell will ultimately lead to the formation of cytokines. And cytokines are the messengers between the cells. Which type of cell has to be recruited, whether it's neutrophil, macrophage, it's the T cell, it's the B cell, which type has to be recruited for the neutralization of a particular type of. Cytokines are basically low molecular weight proteins. They are involved in the initiation and progression of inflammation, and they are released by the epithelial cell. The first cytokines to be produced in the inflammation of the gingival tissues is IL-1, beta, and IL-6, and they are also important from the bone resorption point of view. Another thing, which cytokine you need to remember is TNF-alpha. This is an important cytokine which leads to cell migration, upregulation of adhesion, upregulation of IL-1, beta, and 6, and destruction of extracellular matrix by MMPs and bone cells. So TNF-alpha is important in elaboration of other types of cytokines. It's important in the rankle activation, and we have seen that TNF-alpha deficient mice had less periodontitis. That means it's one of the key cytokines which will lead to the resorption of bone and periodontitis. So what are the functions? They are messengers between the cells. They also lead to angiogenesis. Once the inflammation is resolved, they will lead to the formation of more blood vessels in that area and healing. Cell proliferation, apoptosis. Once the cells have done their job, they need to be taken care of and they need to be eliminated from the body. Tumor metastasis and host defense. So these are different functions. Now you must have come across these different types of classification: CXC, CX3C. What are they? They are the classification system of these cytokines. If C and C, the carbon moieties, are together, they are called as CC. If they're separated by distance, this is called CX3. How much distance, how many X's are there, that will decide what. So these nomenclatures are important from the point of view that how is the structure of these chemokines of the cytokines. So this is, if you see CCl5, CCl2, you should know that CC, if they are together, that means there is no difference, there is no distance between the two C residues. So this is the different types of cytokines that are released, and they're elaborated by all type of cell and they affect all types of cells. So what does the cytokine, you have already gone through this, how this transmigration takes place. Cytokines are important from that point of view. Another cytokine, what you have to remember is IL-8. This is important from the neutrophil recruitment point of view. So this is a chemokine which is produced by gingival fibroblasts, gingival epithelial cells and endothelial cells, and this is the cytokine which recruits the neutrophils to the locally. And another is macrophage chemotaxis protein. Now we have studied about IL-1 beta, we have studied about IL-6, we have studied about IL-8, we have studied about TNF-alpha. These are some of the important things. And macrophage chemotaxis protein is another cytokine which is important from your point of view. Their cytokines, chemokines are normally also produced in the body and they have physiological functions. But when they are elaborated or secreted in immense quantities, then they will lead to the destruction of those cells. So there are IL-4 proliferation of T cells and immunoglobulins are secretion by it downregulates 1 beta. So what is IL-4? It's an anti-inflammatory. IL-4, IL-10 are the anti-inflammatory mediators, and IL-1 and 6, you remember, are the pro-inflammatory mediators. So IL-4 is downregulating IL-1 beta and IL-6 MMPs, and it also induces production of IL-10, which is again an anti-inflammatory mediator. These are the different types of cytokines. So by their nomenclature, now you know how they are classified, why they are called as CCX, CXR, and on what cells they are, which cells produce them, and which cells are affected by them. This is a chart of those. Now we come to the lipid mediators of inflammation. You must have read about that. I'll go briefly through that. So the phospholipid on the cell surface, it is the phospholipase A2 acts on that, the phospholipid. It leads to direct ionic acid metabolism, and there are two types of elaboration, cyclooxygenase and lipoxygenase pathways, which leads to different. Why are we studying these lipid mediators are that they also recruit different types of cells, and PGE2 is mainly important, that is mainly located in macrophage-like cells and is a potent stimulator of alveolar bone cells. Earlier, we used to say that there is OAF was a term, osteoclast activating factor, which led to the bone destruction. That same OAF was later designated at IL-1. So IL-1 and PGE2 are important from the bone resorption point of view. You need to remember that. So PGE2 is also important in the regulation of inflammation. So this is an overall picture of how these different types of cytokines are elaborated, right from the epithelial cells to the fibroblasts to the macrophages, and ultimately lead to the destruction of the bone. Now, destruction of periodontal tissues, it's divided into two types. One is the destruction of bone and destruction of extracellular matrix. First, we will study about this because you get a short note on osteoimmunity also. So it's a field which has emerged about 40 years ago. Earlier, we used to say, as Dr. Ganesh Puthu also said, that the bone destruction in periodontitis is basically a degenerative process. But now we know that it's an active process where osteoclasts and osteoblasts take place, take care of the formation and degradation or the resorption of the bone. And it has been investigated, osteoimmunity has been investigated in different types of bone metastasis, rheumatoid arthritis, and periodontitis also. So what happens that you should remember two things in that. One is osteoprotein and another is rancor, receptor activator of nuclear kappa factor B ligand. Now, the ratio of osteoprotein and rancor will decide whether there will be bone resorption or bone formation. If there is more of the osteoprotein, this will stop this rancor from attaching to the rank, the receptor on the strobel cells, and it will not lead to the formation of active osteoblast. If there is less of osteoprotein, this site open for the attachment of rancor, and this will ultimately lead to the formation of osteoclast, which will lead to the bone destruction. So this is the ratio of osteoprotein and rancor which will decide whether the bone formation is there. So in the initial phases of inflammation, when the macrophages are there, they will elaborate this rancor and there will be bone destruction. And in the later phases of the resolution of inflammation, when osteoprotein is there, it binds with the receptor and it will not lead to the formation of osteoclast. It will lead to the formation of osteoblast, which will form the bone rather than dissolve the bone. So this is the osteoimmunity. In hemostatic conditions, there is a proper ratio of rancor. Rancor is always less than the osteoprotein. In the inflammatory condition, rancor is always greater than the osteoprotein. This will lead to the bone resorption. So this is the ratio between the two, which is important from there. So extracellular components, you know that most of the extracellular component is the collagen, or there are elastins and there are gelatinases, which are different types. They have been divided into different types, and other matrix metabolites, there are two components of the system. One is the organic, another is inorganic. Inorganic is found in the bone, which also has some organic portion. So you need, for the bone destruction, also you need matrix metalloproteinases, and you also need acidic environment or osteoclast, which creates the acidic environment for the resorption of bone. For the collagen degradation, you need the matrix metalloproteinases. So matrix metalloproteinases are there, which have been divided into collagenases, gelatinases, strombolises, and membrane type, and they have physiological roles. They are normally found in the body, and they are also found in the inactive form. So they have a physiological role of maintaining the body system, and pathological roles are there, where they are elaborated in excessive amounts and their inhibition is not. So there are different types of, from the periodontal point of view, you should remember MMP1 will degrade these different components, interstitial collagen, extracellular matrix components. MMP9 will degrade extracellular matrix, basement membrane. 2 is expressed in inflamed pocket and helps in epithelial migration. 13 is important from type 1, 3, and 4 collagen. And MMP7 degrades fibronectin, laminin, and type 4. So basically all these MMPs will take care of all the components that are found, the basement membrane, the types of collagen, the laminin, fibronectin, and collagen. So they will degrade different types of this. So this is a classification, and their substrates, what different substrates they act upon. These are different types of classifications of the MMPs. And how does this MMP activity is regulated? Now you cannot let these matrix metalloproteinases go on degrading the enzyme. So there are tissue inhibitors of matrix metalloproteinases which will inhibit, they will bind to the active site of the enzyme, and they will not let it degrade, or they will not let have its active domain which will degrade the different types of collagenases or proteins. So this is the regulation, the elastins, the different types of proteases. TIMP1, this will tissue inhibitors of metalloproteinases, this will inhibit MMP1 and MMP9, and this is the regulation of that. This is an elaborate mechanism of what is happening at from the innate point of view and adaptive point of view, how activation of antigen presenting cells are there. There are hyperactive monocytes, naive T cells. They are elaborated into different types. How these cells are actually activated depends upon the cytokine milieu. The milieu decides which type of cell will be activated, and accordingly interferon is activated or TGF-beta is activated. And ultimately, if there is a resolution of inflammation, there will be repair. Otherwise, this process will go on, like what I was telling you about the polyclonal B cell activation, and there is unlimited damage and it cannot be stopped. And that is what I told you in the initiation of this presentation, that there is that immunostat which is hypersensitive, where you cannot control the periodontitis. So now we come to the last section, that is the resolution of inflammation. So if we look at periodontitis, basically it is that if there is a normal inflammation, it will go and it will go to the resolution phase. It is the chronic inflammation which does not go into the resolution phase. There is the local microbes are never eliminated from that area because they are endogenous and they cannot be eliminated from that area. You cannot have sterile mouths, and so they will go to the chronic inflammation phase. So this is called as the hypersensitive immunostat, which will go into the chronic inflammation. So what do, there are different agents that have been used, that have been taught to you by the host modulation, that if you use lipoxins, resolvins or protectants, what they do, they will decrease the neutrophil infiltration. They will actually cause apoptosis of the neutrophils which are present there, uptake of apoptotic neutrophils, and they will clear the area so that repair can process. Once the resolution is there, then the fibroblast will elaborate different types of collagens and repair process can be initiated, which is also initiated by the thromboxanes or TGF. Different types of cytokines are also involved in the repair of the tissues. So I want to conclude by saying that understanding of host response in periodontal disease is difficult, but it's a must, because you need to understand the pathogenesis of the disease and different types of mechanisms by which you can stop. So interactions between host immune and pathogen is a complex process which has many components: cytokines, complement, B cell activation, T cell activation, different types of B cell, different types of T cells. And it is important, both B and T cells have an important role to play. And with the present knowledge, it's now clear that the activation of the B cell causes survival, proliferation, inactivation, elimination. It has different types of actions, B cells. And this information can help us in generation of useful diagnostic and even therapeutic. Why we are studying this complex is because we need to generate different types of useful diagnostics and even therapeutic interventions for different types of studies. Thank you. You can contact me on that. And I have tried to compile some questions that have come. This is there in the presentation. So I found 54 questions that have been asked in relation to that. So that's why I went into the details of each component, because then you have short notes on complement, you have short notes on inflammasomes, you have short notes on the antibody, antigens. So I think this presentation should help you in answering all of these questions. Thank you so much.
Thank you very much, ma'am, for a very wonderful, elaborate lecture. Even it was a learning experience, not only for the students, but many of the life members for sure, because this is the one topic which everyone is a bit scared of. And thank you for making it very simple and understandable. Ma'am, if you permit, we'll take a few questions.
Right, sir. Sure, sir.
Students, some have asked about what is inflammasome. I think you have covered it, but if you can just...
I have covered that in my... I know there are many questions asked on inflammasome, so I have covered that. It's a complex of 3-4 proteases which ultimately leads to the formation of cytokines.
Ma'am, can you tell us the clinical application of histocompatibility complex and how does macrophages present antigen to T cells?
This also I have elaborated, but I will again say that there are two types of MHCs, MHC1 and 2. One is found in most of the cells, so they are associated mostly with the elimination of the cells which have been damaged. So MHC1s and MHC2 are only found on the immune cells, which are important from the point of view that they will ingest the microorganisms and utilize. So this is the two types.
Next question is, what is pan receptor defect?
Pan receptor defect. So this and the B cell polyclonal activation is the same. When the receptors are, when there is so much proliferation and formation of antibodies that they do not actually neutralize, they only cause damage. It's like that two-sided sword. On one side it's trying to kill the bacteria, on the other side it's getting its own host tissues damaged. So from that point of view, we see, just a second, there is,
yes ma'am,
the battery is low. So pan receptor damage is basically if the receptors are this is a genetic problem that the receptors are not able to take up the different types of these antigen presenting cells receptors, and they cannot be eliminated by the body.
Next question is, ma'am, what is meant by T cell priming which leads to T cell differentiation?
This is same what I was telling you. The dendritic cells, they lose their cohesiveness and they come out from the epithelium. They go to the local lymph nodes, and then they will stimulate a particular colony of T cells, which will prime them towards that particular elaboration of that particular type of receptors.
Next question is, ma'am, immune cell activity in dormant and progressive lesions of periodontitis.
This I've been repeating in every slide, that it's the local milieu which will decide whether this will go into a more progressive region or it will be taken up by the body. And for that, you need to remember about the immunostat. That's what I was telling you. Either it's insensitive, it's sensitive, or it's hypersensitive. Hypersensitive means that nothing can stop it. And that is refractive periodontitis.
Ma'am, again, someone has asked, what are the few authors which they can quote in this context?
I have given 3-4 those presentations in that you can refer to them for the authors. I think the one I was telling you that I have read recently, 2016 article by COG, that is important from that one.
Thank you, ma'am. Last question for the day. How much is the Rankel and OPG ratio?
It should be 1:1. If it's 2:1, then if Rankel is 2:1, then it will lead to bone resorption. If it is the reverse, then it will reach to the bone formation.
There are like flooding of the questions on the today's topic, ma'am. But I request all the students, because of shortage of time, paucity of time, we cannot take up all the questions here. But definitely they can forward the questions.
Yeah, of course. I have given my email address. They can definitely contact.
Ma'am can definitely solve your questions. Thank you very much, all the ma'am, for wonderful lecture and leading in this lecture series from the front. And because of your leadership only, this all lecture series was possible for us to do. And it was really a very learning experience for all the postgraduate students, ma'am. Thank you very much to you. Thank you very much to our secretary, Dr.
All of you are doing great, sir.
And all the EC members and all the members of Indian Society of Odontology. And we will definitely see you all for a next lecture on Saturday on 30th. I will share you the details for that. And we'll signing off from today. See you in the next lecture. Thank you. Thank you very much all. Thank you, ma'am.